Hello everyone, welcome to my channel Anatomy by Dr. Vijaya. Today's topic will be about cerebellum. So this I'll be doing in two parts, cerebellum part 1 and cerebellum part 2. So in part 1 I'll be covering the anatomical divisions, the structural organization, the microarchitecture of the cerebellum, and also it includes the blood supply of the cerebellum. In the second part, I will be covering the major pathways, the functions and disorders related to the cerebellum. Let us learn about the cerebellum. So this is the first part of the cerebellum where I will be focusing on the structure and general organization of the cerebellum. So this is the sequence of the lecture. So first is the general organization of motor system, so how cerebellum will be influencing the activity in the central nervous system. So we will be learning about that. Then we will go into the gross anatomy of cerebellum, then the topographic map of the cerebellum, arterial supply, and the microarchitecture and the main neurotransmitters which are helping in the activity of the cerebellum. Let us know about the general organization of the motor system. So we all know that motor system, that is the voluntary movements, is mainly influenced by the descending tracts which are coming from the cerebral. Main one is coming from the cerebral cortex, so that's called as a pyramidal tract or corticospinal tract. Other than that, there are extrapyramidal tracts as well. So these tracts mainly influence the voluntary movement. So these are mainly controlled from the cerebral cortex and other parts, so mainly from the cerebral cortex. So whenever an individual executes an voluntary movement, so that influence, that activity where there will be contraction of the muscles, so that is mainly affected by the information received through the sensory system. So as I mentioned, there are general sensation, so the person will act according to the sensation he has received. So it might be a pain, touch, or temperature, or a joint movement, or it can be through vision, eyes, that is through eyes, or smell, or hearing, or sometimes it can be a muscle as well. So according to the influence coming from the sensory system and other special sensation, so the individual executes an action. So there will be voluntary movements where there will be voluntary contraction of the muscles. So this influence is through the descending tracts, okay, so which I mentioned, it is mainly coming from the cerebral cortex, so that's called as corticospinal tract, and there are other tracts as well, so which are supraspinal. So these tracts, that is supraspinal, that is they are above the spinal cord, so either they are coming from the brain stem, so those are categorized under extrapyramidal tracts, which include cerebrospinal, tectospinal, olivospinal. So all these are extrapyramidal system. So all these influence the activity of voluntary movements. So if you see the corticospinal tract, so they mainly influence the highly skilled movements, and the other supraspinal or extrapyramidal tracts, so they mainly influence the basic voluntary movements. Along with that, they help in the adjustment of the muscle tone. So these are the main tracts which are coming from the cerebral cortex and other parts of the brain stem. Along with that, there are influence coming from the basal ganglia and also from the cerebellum. So when you see basal ganglia and cerebellum, so they don't have a direct influence. So if you can see the information coming from the motor cortex, so mainly the precentral gyrus and other premotor areas, so those are the motor cortex, which mainly influence the primary voluntary movements. Along with that, there are extrapyramidal system, so they influence directly, and which mainly sends the information to the lower motor neuron, which are present in the spinal cord. So they have a direct influence. But if you see the basal nuclei and cerebellum, so they don't have a direct connection where they influence the lower motor neuron, but indirectly they help in the smooth contraction of the muscles, okay, and unconsciously they mainly maintain the muscle tone, and they also help in the coordination of the movements, okay. So unconsciously they help in the contraction of the muscles, unconsciously they help in the maintenance of the muscle tone, and also they help in coordinating the movement so that the movement occurs smoothly and also in a finer way, okay. So this is how the motor system functions. So there are stimulus which directly influences the lower motor neuron in the spinal cord, and some influences indirectly. So directly are from the motor cortex and other descending tracts, okay, other which are coming from the brain stem, okay, from the reticular formation. So these are the direct influence on the lower motor neuron. Indirectly is through the basal ganglia and cerebellum, which mainly helps in maintenance of the muscle tone, posture, coordinating the movement, okay, and unconsciously helping in the smooth contraction of the muscle. So whenever an action has to be executed, so there should be contraction of the group of muscles for that particular action, and there should be relaxation of the antagonist muscles. So these are mainly coordinated by the basal ganglia and the cerebellum, which mainly sends information to the brainstem and also to the cerebral cortex, and then the descending stimuli are made finer so that the movement occurs in a better way. So cerebellum, it is a part of the hind brain, so it is the largest part of the hind brain. So you know that embryologically the central nervous system is divided into forebrain, midbrain and hind brain. So hind brain includes medulla oblongata, pons and cerebellum. So among that, cerebellum is the largest part in the hind brain, okay. So they are located behind the pons and the medulla. So here is the pons and this is the medulla. So here you can see the sagittal section of the brain, okay. So this is pons, this is medulla oblongata, and here you can see the fourth ventricle. So behind pons and medulla, this is the cerebellum, okay. So compared to the sulci and gyri, even the cerebellum is folded in order to occupy more neurons, and compared to cerebrum, cerebellum has more number of neurons, okay. So they say that 80% of the neurons are mainly present in the cerebellum when compared to cerebrum. So around 50 billion in cerebellum and around 20 in the cerebrum, okay. So since there are more neurons within a limited space, so even cerebellum is folded, okay. So if you can see, if you compare the sulci and gyri with the fissures and pleats, cerebellum will be more folded, okay. So in order to occupy more neurons, okay. And when you can see here, this is the superior surface of the cerebellum. So they are separated from the occipital lobe. So here is the occipital lobe of the cerebrum. So they are separated by a dural fold called as tentorium cerebelli, okay. So the tentorium cerebelli is situated here, which separates the cerebellum from the occipital lobe of the cerebrum. So let us know about the divisions of the cerebellum. So cerebellum can be divided into multiple ways. So there are anatomical divisions, there are functional divisions. So anatomical divisions of cerebellum is, they are divided into 3 lobes: anterior lobe, posterior or middle lobe and floccular nodular lobe. So if the cerebellum is opened up, okay, if you can see, the cerebellum is looking like this, okay. So this is the superior surface of the cerebellum and this is the inferior surface of the cerebellum. So when you open it, you can see for the descriptive purpose, so if you open the cerebellum, you can see the lobes, okay. So this part is the anterior lobe, so and this whole thing is the posterior or the middle lobe, okay. So the superior and the inferior surface of the cerebellum can be separated by a fissure called as horizontal fissure. So there is a deep fissure which separates the cerebellum, not completely, incompletely. It separates the cerebellum into superior surface and inferior surface, okay. So this is the horizontal fissure, you can imagine. So this is the superior surface and this is the inferior surface. Okay. On the superior surface, you will be seeing anterior lobe here, and then you will be seeing the part of posterior lobe. So posterior lobe or the middle lobe, it occupies both superior and inferior surface. Okay. So partly it is present on the superior surface and the majority it is present on the inferior surface. So anterior lobe is separated from posterior or the middle lobe by a fissure called as primary fissure. So why it is called primary fissure? Because it is first to appear or first to develop in the embryological period when during the development of cerebellum. Okay, so that's why it is named as primary fissure. Okay, so you see other fissures as well, but why these are named as fissure? Because it is quite deep when compared to other sulci. Okay, so that's why they are named it as fissure. So the first fissure you see primary fissure, which separates anterior lobe of the cerebellum from the posterior or the middle lobe. As I mentioned, posterior or the middle lobe is present throughout from superior to inferior surface. And when you go to the inferior surface, so there is one more lobe. Okay, so this lobe is called as flocculonodular lobe. Okay, so this flocculonodular lobe is separated from posterior lobe by a fissure, very deep fissure, that's called as uvulonodular fissure. Okay, so which you can see here. So uvulonodular fissure separates flocculonodular lobe from the posterior lobe. Okay, so this is the anatomical division of the cerebellum. So three lobes: anterior, posterior, also called as middle, and flocculonodular lobe. Anterior is separated from middle or posterior by primary fissure, and posterior or middle lobe is separated from flocculonodular lobe by uvulonodular fissure. Uvulonodular fissure. And you will be seeing one more fissure which separates the cerebellum into two surfaces, superior and inferior. So that fissure is called as horizontal fissure. Okay. So this is the anatomical division. Then, when you see the cerebellum, it has two hemispheres, right and left, and the two hemispheres at the center, they are joined by a structure here called as a vermis. So this center middle part is called as a vermis, and on either side are the hemispheres. So they are called as cerebellar hemispheres. Okay, so two right and left cerebellar hemisphere are joined at the midline by a structure called as vermis. Okay, so functionally, the cerebellum is divided into the middle part, that's called as a vermis, paramedian or a paravermal area, and lateral area. Okay, so three areas. This is a functional division. So vermis at the center, paramedian or intermediate or paravermal, all are same. Okay, and lateral hemisphere, and there is one more functionally, so that is the oculonodular lobe. It is categorized under separate division functionally, so that's called as vestibulocerebellum. Okay, so you can see the functional division: vermis, the vermal zone or the vermis, paravermal zone of hemisphere or paramedian, then you have the lateral zone of hemisphere, and the flocculonodular lobe, it has a different function which is related to the vestibular system. So that's called as vestibulocerebellum. You can see this is the vermal zone, then you have a dotted one, paravermal zone, and most closely packed dotted zone, that's the lateral zone of hemisphere. So vermis, so it is considered as spinocerebellum because it mainly gets input from the spinal cord. Okay, so that's why it is called as a spinocerebellum. So the function of, as I mentioned, this is the functional division. So the function of vermis is it mainly helps in maintaining the posture, that is it has influence on the the truncal and the proximal area. So it mainly has influence on the truncal part of the body and the proximal, the limb part of the body, the limb girdle. So if you see again the limbs has the distal part and the proximal part. So it has influence on the proximal muscles and the truncal muscles, okay, to maintain the posture. So their main function is to maintain the posture and maintain the tone of the muscle, okay. So their input is mainly from the spinal cord. So they are categorized under spinocerebellum. So whenever there is lesion which is mainly affecting the vermal area, the vermis area, so definitely the patient will have difficulty in maintaining the posture. So they have truncal ataxia, okay, postural imbalance, and this is more the anterior part. If you can see, this is the vermis. So the anterior vermis, it's mostly affected when a person is addicted to alcohol, okay. So if he is very much alcoholic, very much addicted to alcoholic, so they are more susceptible to lesion in the anterior vermis. So they will have signs and symptoms of truncal ataxia and postural imbalance. So ataxia means a loss of control on the body movements, okay. So there won't be a coordinated movement, okay. So there won't be control in the posture or the tone, and there will be postural imbalance, okay. So this is seen whenever there is lesion affecting the vermis zone. The next is paramedian or paravermal or intermediate hemisphere. So they are also categorized under spinocerebellum because even they get input from the spinal cord. So this part mainly has influence on the distal muscles, okay. So they also help in maintaining the posture, but they have more control towards the hands and feet, the distal part of the muscle. So if you see the, as I mentioned, limbs, the proximal part, upper limb, if you see the shoulder region, pectoral region are considered as a proximal part. Then the forearm and the hands, mainly the hands are considered as a distal part, okay. So vermal area mainly maintains the posture and tone on the truncal area and the proximal, the girdle area, okay. So in the upper limb it is mainly pectoral girdle, shoulder girdle and part of arm as well. And in the lower limb it is mainly the hip area, the gluteal region. So all those are considered as the proximal part. And intermediate, that is the paramedian area, so it maintains the distal, so that is mainly the hands and feet, okay. So it maintains the posture and tone of the distal area of the limbs. So whenever there is a lesion, so the patient will present with lesion in the paramedian. Again there is loss of body control on the movement, so there will be loss of coordination, there will be loss of tone, okay. So that's called as limb ataxia. So here it is mainly on the trunk, okay, at the center part, the truncal and the proximal part, but here it is mainly seen on the distal area. So limb ataxia, and usually you can see intention tremor. So whenever you ask the patient to pick up something, or you can place an object, so this is one of the tests they do whenever in the CNS examination, whenever you are suspecting any cerebellar lesion. So you will keep an object and ask the patient to reach towards that object. So whenever he or she has to execute the action, they will have a shivering hand, okay. So that's called as the intention tremor. And they also do a test called as diadochokinesia, okay. So where they do alternate movements. Very good example. Usually during the CNS examination they do supination and pronation, okay. So you ask the patient to do this. So the individual who is having a lesion over the paramedian part of the cerebellum, so they'll have dysdiadochokinesia. So they'll have difficulty in executing this action, okay. So they can't do supination and pronation, okay. Means they'll have difficulty in coordinating that, okay. So that condition is called as dysdiadochokinesia, okay. Limb ataxia, intention tremor and dysdiadochokinesia. The third hemisphere, that is the lateral hemisphere. So this is called as cerebro or neopontocerebellum, because they are principal input is from the cerebral cortex. So they mainly receive information from the cerebral cortex. They pass through the pons and then enter the cerebellum. So that's why it is called as neopontocerebellum. So if you can remember, there are there are corticopontine fibers which are coming from the cerebral cortex. So they come from cerebral cortex, goes to the pons area, and from pons there are transverse fibers from the basilar part of the pons which enters the cerebellum. So if you can go through my video on the brain stem, that is the pons, so where I have mentioned there are these fibers coming from the cerebral cortex. So these mainly go to the lateral hemisphere of the cerebellum. So their principal input is from the cerebral cortex. So their main function is, again, it has the overall planning and execution of the movements, coordination. Okay, so it gives mainly feedback to the motor cerebral. So whatever information it receives from the cerebral cortex, in turn, as I mentioned, cerebellum and basal nuclei doesn't have direct influence on the lower motor neuron. So they influence the lower motor neuron indirectly by sending the information to the cerebral cortex and to the brain stem. So whenever the movements has to be corrected, whenever the movements has to be made finer and smoother, so that there is a correct execution or a proper execution of the movements. So in order to correct those movement errors, the information again have to go back to the cerebral cortex. So it gives feedback to the motor cerebral cortex to correct the movement error, so that whatever the movement is executed is coordinated and it occurs in a smoother and a finer way. Okay, so it has the overall control on the coordination, planning and execution of the movements. So whenever there is a lesion in this, again the patient will have ataxia. So it can be limb ataxia or truncal ataxia because it has the overall control. Okay, so there is no definitive disorder associated with localized restricted lesion. So you can't tell that this is from the lateral hemisphere, because since it has a overall control on the execution of the movement. So the patient might present with, even with intention tremor, they might have ataxia, truncal and limb ataxia. Okay. The fourth functional division, which is the vestibulocerebellum, the lobe which conducts this function is the flocculonodular lobe. So this is interconnected with the vestibular nuclei. So their main function is to control the posture, the head movements, the balance, coordinated eye movements. So all these are controlled through flocculonodular lobe. So they get input and the output goes to the vestibular nuclei. So whenever there is a lesion affecting this lobe, so mainly it controls the eye movement, coordinated eye movements. So the patient might present with nystagmus, rhythmic oscillation of eyes, which is involuntary. So there will be involuntary rhythmic oscillation of eyes. Then they have difficulty in balancing. So you know that vestibular nerve and the nuclei are mainly related with the balance. So they mainly control the balance. So since the patient with the lesion in this flocculonodular lobe, they have difficulty in balance. So whenever you ask the patient to walk, so they might have, in order to maintain the balance, so they might have a wide ataxic gait. Okay. And also the patient tends to fall, so they have difficulty in balance, so they tends to fall towards the affected side, because the cerebellum mainly controls the same side of the body, in contrary to the cerebral cortex. Cerebral cortex maintains the body on the contralateral side, but cerebellum controls the body on the same side. Okay, so right cerebellar hemisphere controls the right side of the body, and left will control the left side. So it is on the same side. So the patient tends to fall towards the affected side. So these are the functional division. So we have learned about the anatomical and functional division. There is one more division according to by the evolution. So if you see, archicerebellum is the most oldest one, which is very well developed in birds. So they are mainly in relation to the vestibular part. So they mainly help in, the birds fly, so they're able to so it has to be well developed so that they can maintain balance while flying. Okay, so they say that it is very well developed in birds. The next to the evolution is the paleocerebellum. So paleocerebellum, it's related to the spinocerebellum. Okay, so where it is very well developed in the terrestrial animals like lizards, crocodile, where they use their limbs. Okay, then the next as the evolution went on, the latest one is the neocerebellum, which is very well developed in human beings. So you have fine movements. Okay, so mainly the skilled movements is through your fingers. So that's why neocerebellum is the most latest one, and they are higher in evolution, and they are very, very well developed in human beings. So this is the different multiple ways of divisions of cerebellum. So mainly you have to know the anatomical division and the functional division. So the red one, archicerebellum, the vestibular cerebellum part. Then paleo is the spinocerebellum part, which includes vermal area, the vermal zone, and also part of paravermal. And neocerebellum is mainly the lateral hemisphere and also the paravermal or intermediate. So this is the topographic representation of our body, the components of the body on the cerebellum. So if you can see the trunk, the axis, which includes the neck, the chest area, the abdomen area, the shoulder, arm, hip, thigh. Okay, so it is mainly represented on the vermis, so which you can see here, the trunk axis, and the proximal limb. As I mentioned, proximal limb mainly includes shoulder and arm in the upper limb, hip and thigh in the lower limb. So they are mainly represented on the vermis area. Then distal extremities, which includes forearm and hand in the upper limb, then the legs and the feet in the lower limb. So they are represented, they are mapped on the paramedian or the intermediate regions. Okay, so there is no distinct map. So this is how it is represented. And lateral hemisphere, overall they are concerned with the planning and coordination and the execution of the movements, and they correct the errors. Okay, so this is how the representation of our body on the cerebellum. Coming to the blood supply to the cerebellum, so they are mainly supplied by the branches coming from vertebrobasilar artery. Okay, the two vertebral, right and left vertebral artery join to form basilar artery. The branches from this both vertebral and basilar artery supply the cerebellum, so which includes superior cerebellar artery and anterior inferior cerebellar artery, which are coming from basilar artery, which you can see here. Superior cerebellar, anterior inferior cerebellar, they are mainly branching from basilar artery. And posterior inferior cerebellar artery, so which mainly comes from vertebral artery. So these are the three main branches which supplies the cerebellum. So there are color coordination. So if you can see, superior cerebellar artery, the green one, mainly supplies the anterior lobe. Okay, and also part of the posterior lobe on the superior surface. And the red one, posterior inferior cerebellar artery, mainly supplies the inferior surface of the cerebellum. Okay, and the rest is by anterior inferior cerebellar artery. So this is the blood supply to the cerebellum, the arterial supply to the cerebellum. Whenever there is a block in the vascular territories, so most common is superior cerebellar artery and posterior inferior cerebellar artery. In the brainstem, you have heard the Pica syndrome, posterior inferior cerebellar artery, which also supplies the medulla oblongata, the lateral area of the medulla oblongata. Okay, so these are the two common arteries which gets blocked. Okay, and that leads to neurological deficit. So mostly superior cerebellar artery infarct presents with pure cerebellar signs because it supplies most of the cerebellum. But if you see posterior inferior cerebellar artery supplies even the brainstem, mainly the medulla oblongata. So the patient might have a mix of cerebellar signs and symptoms and also lateral medullary syndrome signs and symptoms. Okay, so the pure cerebellar signs are mainly seen whenever there is infarct of the superior cerebellar artery. So posterior inferior and anterior inferior cerebellar artery have sensory and motor deficits because they also supply the brainstem. So there will be involvement of brainstem as well. So let us move on to the microarchitecture, that is the histology of the cerebellum. So as you all know that cerebellum and cerebrum has outer grey matter which is called as the cortex, then inner white matter, and innermost there are some nuclei. Okay, so similarly, that's what even in the cerebellum it has the cortical area, that is the grey matter area, has 3 layers. So 3 layers in the cortex area, that this is the grey matter area. So they are from outside, from outer to inner or superficial to deep. So you can see molecular layer, then a layer of Purkinje cells, and the granule cell layer. So these are the 3 main layers present in the cortical part of the cerebellum. Okay, cerebellar cortex. Then you see the white matter, where you can see the dendrites and axons of the neurons. Okay, and the innermost you will be seeing some nuclei. So they are called as intracerebellar nuclei, the intercerebellar nuclei. They are 4 in number. Okay, so here you can see this is the outermost molecular layer. So mainly has axons and dendrites. Okay, so they have the dendrites of the Purkinje cells and axons of the granule cells which are distributed here. So within that axons and dendrites, you will be seeing lots of other neurons as well, that is the stellate cells, the basket cells. So all these are distributed in the molecular layer. So molecular layer has a wide arborization, that is the branches. So mainly the dendrites of the Purkinje cells, which Purkinje cell is a separate layer here. Okay, a single layer of Purkinje cells. So their dendrites branches into the molecular layer, and the axon moves into the granule cell layer. And the granule cells which are present in the deepest part, it sends axons to the molecular layer. So you will be seeing lots of axons and dendrites over the molecular layer, and along with that there is distribution of other neurons. Those are stellate cells and the basket cells. So these stellate cells and basket cells, they have small branches which will have a synaptic contact with the dendrites of the Purkinje cells, which you can see here. Okay, so they have synaptic contact with the dendrites of the Purkinje cells. So they influence the activity of the Purkinje cells. The next is the Purkinje layer, cell layer. As I mentioned, there are large flask-shaped the Golgi type 1 neurons. Okay, so they have a wide branching of dendrites which moves to the molecular layer, and a long axons. Okay, which mainly has a synaptic contact with the intercerebellar nuclei. Okay, so the nuclei which are deepest in the white matter area of the cerebellum. Okay, the next third layer is the granule cell layer. So here you can see mainly the important cell is the granule cells. Along with that there are other fibers as well. So you will be seeing mossy fibers and climbing fibers, and there is one more neuron which is called as the Golgi cells. Okay, so these are mainly seen in the granule cell layer. So this granule cell, this is the granule cells. They have dendrites. Okay, so the dendrites are present in the same layer, so they mainly have synaptic contact with the mossy fibers. Okay, and the axons of these granule cells move into the molecular layer, which will have a synaptic contact with the Purkinje cells. Okay, and this is the Golgi cells. even they have dendrites and axons which have a synaptic contact within the granule cell layer. Okay, so they don't have a contact with Purkinje cells, but they have contact with the granule cell layer and the mossy fibers and also climbing fibers. So what are these mossy and climbing fibers? So you know that cerebellum receives lot of input through the peduncles, which you have learnt in the brainstem, okay, the neuroanatomy of the brainstem. So all the three brainstem are connected to the cerebellum by three main peduncles, the superior, middle and inferior cerebellar peduncles. Okay, so through these peduncles, mainly from middle and inferior, okay, so they mainly have afferent fibers which move to the cerebellar cortex, okay, and they are called as climbing fibers and mossy fibers. So if you can remember, there was a nuclei in the section of the upper part of the medulla, that is at the level of olive. So there was a very important nuclei called as inferior olivary nuclear complex. Okay, so this inferior olivary nuclei sends fibers to the cerebellar cortex through inferior cerebellar peduncle. So those fibers which are entering from the inferior olivary nucleus to the cerebellum, those are called as climbing fibers. So they are nothing but olivocerebellar fibers. Okay, so olivocerebellar fibers are called as climbing fibers. Okay, so these climbing fibers, so they climb. Why they are called as climbing? Because they ascend like a vine. Okay, so there is a plant called as a vine, so which ascends on the tree. Okay, so this also ascends through the white matter, then it enters the cortical layer, then it enters the granule cell layer, then to the Purkinje cell, and then it winds around the dendrites of the Purkinje cells. Okay, so these have influence on the Purkinje cells. Okay, so they release neurotransmitters and influence the activity of Purkinje cells. The next other afferent fibers. So cerebellum also receives fibers through middle cerebellar peduncle, okay, and also other fibers from inferior cerebellar peduncle, which you can see. There are some fibers coming from the spinal cord, which are called as spinocerebellar tract, that is posterior and anterior spinocerebellar tracts. Then they also receive from the pons, that is the information of the cerebral cortex. The corticopontine fibers later form transverse fibers which enters the cerebellum through middle cerebellar peduncle. So those, the other afferent fibers will be called as mossy fibers. Okay, so they have contact with the granule cell, okay, and also with the Golgi cells. Okay, so those fibers are called as mossy fibers. So mossy and climbing fibers are afferent fibers which enters the cerebellum through the peduncles, superior, middle and inferior peduncles. So climbing fibers are mainly coming from inferior olivary nucleus through inferior cerebellar peduncle, and the rest of the afferent fibers are called as mossy fibers. So two inputs, climbing fibers and mossy fibers. Molecular layer contains two types of neurons, stellate cells and basket cells. Purkinje cell, as I mentioned, large Golgi type 1 neurons, flask shaped and are arranged in a single layer. As I mentioned, the dendrites undergo profuse branching and they are mainly seen, the branching of those seen in the molecular layer, and each dendrites will have a spines, okay, which will have synaptic contact with the climbing fibers and with the granule cells. Okay, the axons pass through the granular layer to enter the white matter. Okay, so they mainly synapse with the cerebellar nuclei. Okay, so this is the axon of the Purkinje cell and this is the dendrites. The axon terminates by synapsing with the cells of one of the intracerebellar nuclei, okay, within the cerebellum, within the white matter of the cerebellum. Granular layer contains granule cells, Golgi cells and cerebellar glomeruli, that is the connection between the Golgi, granule and the mossy fibers. Each granule cell gives rise to 4 to 5 dendrites, okay, which have synaptic contact with mossy fiber input. Okay, so you can see here, so this is the granule cells, okay, so they have dendrites which have synaptic contact with the mossy fiber. So this is the mossy fibers. Okay, so this connection between the mossy fibers, the dendrites of granule cells and the Golgi cells, the axons of the Golgi cells, so that is considered as a glomerulus. So this is Golgi cells. Golgi cells is a little bit bigger, so their dendrites go into the molecular layer, okay, but they have axons which have a connection with the mossy fibers and the granule cells. So that connection will form the glomeruli. Each granule cell gives rise to 4 to 5 dendrites which have synaptic contact with mossy fiber input. Axons of each granule cell pass into the molecular layer. Okay, so they form a very long axon, then they form a parallel fibers. They have a T-shaped bend. Okay, so this you can see here, this is the red one, represents the axons of the granule. Okay, so once they reach the molecular layer, they form a T-shaped parallel fibers which have a contact with the synaptic contact with the Purkinje cells. Okay, axons of each granule cell passes into the molecular layer forming parallel fibers which run at right angles to the dendritic process of the Purkinje cells. Then the dendrites of the Golgi cells ramify in the molecular layer and their axons terminate by splitting up into branches that synapse with the dendrites of the granular cells. Okay, so if you can see here, the dendrites of the granular cells will have a synaptic contact with the mossy fibers. Okay, the axons of the granule cells, they pass to the molecular layer, okay, and they form a T-shaped parallel fibers which have synaptic contact with the Purkinje cells. Then the Golgi cells which are present in the, which you can see here, this is a Golgi cell. So their dendrites pass to the molecular layer and the axons, they undergo branching which will have a synaptic contact with the granule cells. Okay. So when you see the two inputs, climbing fiber, the one is the climbing fibers which are afferent excitatory fibers. So they mainly they release neurotransmitter which stimulates, which excites. Okay, so they are mainly coming from the inferior olivary nuclei. So they are called as olivocerebellar tract. So when you see, they are like snipper-like, they are highly specific. So they mainly have synaptic contact with the dendrites of the Purkinje cells which are present in the molecular layer. Okay, each climbing fiber are dedicated to one Purkinje cell. So if you see here, one Purkinje cells have lots of branches of dendrites. So each climbing fiber have synaptic contact with all the dendrites of one Purkinje cells. So they are very highly specific, like a snipper gun which is highly specific. Okay, so they are dedicated to one Purkinje cell. But in between they have small branches also which will have a contact with basket or something, but their main synaptic contact is with the Purkinje cell. The next input, the mossy fibers. So other afferent fibers which are coming from spinal cord, that is spinocerebellar, coming from pons, pontocerebellar, vestibular nuclei, vestibulocerebellar tracts. So all those are grouped together as mossy fibers. So they terminate as mossy fiber rosette. Okay, so they form a rosette here. Okay, so the mossy fiber rosettes on granule cell dendrites. So here this is the granule cell, so which has dendrites in the granular cell layer. So they have synaptic contact with the mossy fibers. Okay, and this is the granule cell axons. As I mentioned, it travels through the molecular layer and they form a T-shaped bend to form, and they run parallelly. Okay, so there is no direct, these mossy fibers, if you see, they have influence on the Purkinje cells but not directly, just like climbing fibers. They don't have direct connection with the Purkinje cells, but they influence the activity of Purkinje cells through granule cells. So the inputs go through thousands of granule cells. So if you can see here, so one mossy fibers can have synaptic contact with many granule cells. Okay, so through thousands of granule cells, they spread, which will spread the activity through parallel fibers to the Purkinje cells. Okay, so these axons of the granule cells have synaptic contact with the spines, dendritic spines present in the dendrites of the Purkinje cells. So the mossy fibers influence the activity of Purkinje cells through granule cells, but climbing fibers have influence the activity of Purkinje cells directly. So they have direct contact with the Purkinje cells, but mossy fibers has connection through the granule cells. So inputs through thousands of granule cells, so thus they spread the activity through parallel fibers into the thousands of Purkinje cells. So if you see, they are compared to a machine gun, which is not very specific, they are highly diffuse, they are divergent. Okay, but climbing fibers are highly specific. So all the outputs from the cerebellum begin from Purkinje cell, right? So the Purkinje cells mainly releases inhibitory neurotransmitter. So the axons of the Purkinje cells mainly have synaptic contact with the cerebellar nuclei. Okay, deep cerebellar nuclei. Okay, so mainly they are inhibitory in nature. So when you see, so the inhibitory action, the inhibitory effect is fluctuating. So it depends upon the stimulation of the Purkinje cells. Okay, the mossy fibers, the climbing fibers, so both are excitatory in nature. Okay, so they release mainly the excitatory neurotransmitter, the glutamate. Okay, so once the excitatory neurotransmitters are released, so that will excite the Purkinje cells, and the Purkinje cells will transmit the stimuli to the, and it stimulates the deep cerebellar nuclei. Okay, but the neurotransmitter released by the Purkinje cell is inhibitory in nature, which is GABA. Okay, so there should not be too much excitation of the Purkinje cells. Okay, whenever there is too much excitation, too much stimulation of Purkinje cells, so what happens? So whatever the output will be always inhibitory. So there is fluctuation of inhibition. Okay, so so Purkinje cells, though they are excited by parallel or climbing fibers, okay, parallel fibers are mainly from the granule cells, okay, which are influenced through mossy fibers. Okay, but the Purkinje cells are inhibited by basket and stellate cells, which are present in the molecular layer. Okay, so there is fluctuating inhibition and excitation. Okay, so in the cerebellum, so according to this fluctuation, the muscular activity is modified and the output goes, the efferent fibers, the information goes to the cerebrum, the cerebral cortex and the brainstem, and accordingly the movements are corrected and they are coordinated well, and the execution of the movement occurs smoothly. Okay, so this is how the cerebellum receives the input from different tracts, different fibers, and through their nuclei, through their neurons, they influence the motor activity, the motor movements. Okay, so that's why whenever you see, though the Purkinje cells are excited by parallel and climbing fibers, and they release inhibitory neurotransmitter which inhibits the deep cerebellar nuclei, but in turn this whatever the excitation which is occurring within the cerebellar environment, okay, so they are controlled, and even the excitation of the Purkinje cells are controlled through other neurons, which is mainly basket and stellate cells. Okay, so they inhibit Purkinje cells, but other climbing fibers and parallel fibers, they stimulate and excite the Purkinje cells. Okay, so as I mentioned, there should not be too much of anything. Okay, so there should be a balance. Okay, so this excitation and inhibition of the Purkinje cells are balanced through these network. Okay, so this is how the muscular activity are balanced and they are coordinated. So there are internucleal neurons as well, that is between the stellate cells, Golgi cells and basket cells. So even Golgi cells, they are rich in GABA, they are inhibitory in nature. So they also influence, as I mentioned, these axons of the Golgi cells have connection with the granule cells, so they might stop the granule cells to excite the Purkinje cells. Okay, and the stellate and basket cells also have connection with the dendrites of the Purkinje cells. So whenever there is overexcitation, so that is again controlled by the neurotransmitters released by the stellate and the basket cells. So this is how the network is balanced between excitation and inhibition. So coming to the deep cerebellar nuclei, which are found within the white matter, and they are mainly represent the output nuclei of the cerebellum. So axon fibers from these nuclei, they exit out, they form the efferent fibers, and the information goes to the cerebral cortex and other areas of the brainstem. So from medial to lateral, the nuclei are named as vestigial nucleus, interposed because they are grouped together, which includes globus and emboliform, and the most lateral one and the largest one is the dentate nucleus. Okay, vestigial nucleus, they are mainly have connection, their input is mainly from the archicerebellum. So they send fibers, efferent fibers, mainly to the vestibular nuclei. Okay, they mainly deal with balance and equilibrium. Globus and emboliform, they are mainly associated with paleocerebellum, that is spinocerebellum. So they mainly maintain the limb posture and the truncal posture as well. So they deal with walking and arm movements. The dentate nucleus, they are most lateral and the largest part. So they are in relation to neocerebellum with the highly skilled movements. So they are associated with highly skilled movements. So these are the deep cerebellar nuclei, which gets afferent fibers from the axons of Purkinje cells, and then they send efferent fibers to different areas of the brainstem, and also sends information to the cerebral cortex. So receives input from incoming and outgoing neurons of cerebellum. So it acts like a toll gate, okay, and modulate the influence of the cerebellum on rest of the nervous system. So this table summarizes the neurotransmitter and the target cells of each neuron. So in the cerebellum, it mainly has the Purkinje cells. So the main cell through which the output and input are influenced is by the Purkinje cells. So their main target is on the cerebellar nuclei. As I mentioned, they mainly release inhibitory neurotransmitter, that is a GABA, so inhibitory in function. So whenever these neurotransmitters are released in the axon terminal, so they have inhibition. The inhibitory neurotransmitters are released to the cerebellar nuclei. So definitely the output will be inhibitory in nature. So granule cells, they have a parallel fibers which have synaptic contact with the Purkinje cells. So the main target is on the Purkinje cells, but they mainly release excitatory neurotransmitter, which is glutamate, which will excite the Purkinje cells. Stellate and basket cells, their target is again on the Purkinje cells, but they are inhibitory in nature because they release inhibitory neurotransmitter GABA. Golgi cells, which are mainly present in the granule cell layer, but their target is on the granule cells. Again, granule cells should be controlled. Okay, so if granule cell is not controlled, it has a more excitation on the Purkinje cells, and the output will be more mainly inhibitory in nature. So as I mentioned, there should be a balance between inhibition and excitation. So Golgi cells will have inhibitory action on granule cells through their axons. This is the division of the cerebellum, the summary. So main anatomical division is anterior lobe, posterior lobe and flocculonodular lobe. Anterior lobe, phylogenetically they are under paleocerebellum. Okay, so functionally they are grouped under spinocerebellum. So they mainly get input from the spinal cord. Okay, and the deep cerebellar nuclei, which mainly controls is the interpose, that is the globose and emboliform. Okay, so posterior lobe, so they are mainly concerned with phylogenetically, they are classified under Neocerebellum. So functionally, they have connection with the cerebral cortex and other areas of the cerebellum. So the nuclei related to this lobe is the dentate nuclei. Okay, the dentate nucleus. Flocculonodular lobe, phylogenetically they are under Archicerebellum. So functionally they come under vestibular cerebellum because they have interconnection with the vestibular nuclei. And the deep nuclei related to this lobe is the vestigial nucleus. So this is the summary of the divisions of the cerebellum. So these are my references. So that completes the first part of the cerebellum. In my next video, that is the cerebellum part 2, I will be covering the major pathways, the efferent and the afferent, their functions and the disorders related to the cerebellum. Thank you for watching.